Welcome to the deep dive. Today we're really getting into the weeds, cutting through a pretty dense stack of sources on immunopathology, autoimmunity.
Yeah, the goal is really to pull out the core mechanistic insights, the things that connect that fundamental T-cell biology we all learned to the kind of challenges we see now. Think COVID, CAR T toxicities.
Right. We want to extract those key clinical takeaways. This is very much aimed at, physicians, researchers in the field.
Exactly. It's about the how and the why for a medical audience. We'll be looking at, well, the checkpoints that are supposed to maintain self-tolerance, the role of key players like IL-6, and how things like viruses and genetics intersect to push towards autoimmunity.
And these connections aren't just academic, right? Our sources are drawing direct lines. Understanding these pathways, it impacts therapeutic decisions every day, like why you'd reach for tocilizumab in a cytokine storm.
Precisely. Or why a specific HLA type might predispose someone to, say, a post-viral syndrome. It gives you that molecular context for managing these really complex, multi-system inflammatory disorders.
Okay, so we're moving beyond just listing symptoms. We're digging into the core failures at the cellular, the genetic level.
That's the plan. Let's start at the beginning. The checkpoints meant to keep T cells in line.
Okay, let's unpack that foundational T-cell biology. Self-tolerance. Where does the system first hit the brakes on potentially self-reactive T cells?
Well, the first major checkpoint is right at activation. A naive T cell, fresh out of the thymus, it's actually pretty resistant to just going ahead and proliferating.
Needs more than one signal.
Exactly. It needs the T cell receptor, the TCR, to see its antigen on an MHC molecule, sure, but it also needs that crucial second signal, costimulation, and that mainly comes through the CD28 receptor interacting with B7 molecules on the antigen-presenting cell.
And if that second signal, the CD28 signal, is missing, which I guess would be typical if a T cell bumps into a self-antigen on just a regular tissue cell, not an activated immune cell.
Right. If you only get signal 1, the TCR signal, without CD28 chiming in, the T cell generally enters a state called anergy.
Anergy.
Meaning it becomes functionally unresponsive. Mechanistically, you need that CD28 input because it strongly boosts the signals going through pathways like PI3K-HKT and, crucially, NF-gov.
Nephews survival signals.
Yes. Without that CD28 boost, activation of these pathways is just insufficient. The cell gets stuck, it becomes refractory, it won't respond properly if it gets stimulated again later. That's a cornerstone of peripheral tolerance.
So anergy stops it from really getting going.
Yeah.
But what if the self-antigen signal, signal 1, is unusually strong? Can it sometimes push the cell to divide a bit, even without costimulation?
That can happen, yeah. But there's usually a built-in safety net, a kind of molecular override.
Okay.
Even if a strong TCR signal alone manages to kickstart some proliferation, that process is typically cut short by apoptosis programmed cell death.
Why? What's the mechanism there?
It comes back to that survival signaling balance, particularly NF-key. Remember, CD28 strongly activates NF-key. Without that strong signal, you get insufficient production of key anti-apoptotic proteins. The big one here is BCL-XL.
BCL-XL, right, one of the BCL2 family members, keeps mitochondria stable.
Exactly. And BCL-XL is a direct target gene of NF-key. So no strong costimulation means weak NFB activation, which means low BCL-XL levels.
And low BCL-XL
means the pro-apoptotic machinery inside the cell wins out. The cell basically commits suicide because it lacks sufficient survival support. It's like the system judges that proliferation without costimulation is too dangerous, too likely to be self-reactive, and eliminates the threat.
A fascinating internal calculation. And this ties into other death pathways too, like FAS.
Yes. The FAS pathway is a really important connection here. It highlights this overlap between maintaining self-tolerance and preventing cancer. FAS is involved in activation-induced cell death, or AICD, which is how the body gets rid of T cells after an infection is cleared, but it's also critical for deleting self-reactive clones.
And the clinical data backs this up.
Absolutely. Our sources point to inherited FAS mutations. In humans, these cause autoimmune lymphoproliferative syndrome, ALPS. These patients have severe multi-organ autoimmunity and a significantly increased risk of lymphoma. You see similar things in mouse models with FAS defects.
Wow.
And it's not just inherited mutations. Somatic mutations in FAS acquired later in life have also been found in patients with autoimmune diseases and in lymphomas. It really underscores FAS as a crucial checkpoint for both immune homeostasis and tumor suppression.
Okay, so that's the cell-level control. But autoimmunity often seems to creep up maybe years after some predisposition is there.
Mm-hmm.
This points towards needing multiple failures, right?
Yeah.
These multi-step models.
That's the key idea. We all generate potentially self-reactive T and B cells just through the random process of receptor diversification. That's normal. The healthy state, let's call it Model A, is when multiple checkpoints anergy, AICD via FAS, regulatory T cells work together to keep these clones suppressed. Autoimmunity happens when these layers of control fail sequentially.
Let's talk through Model B then. This covers inherited genetic risk, which is common.
Right. In Model B, someone inherits a variant, maybe an FAS like we discussed, or maybe in a gene like PTPN22, which sets the T-cell activation threshold. This inherited defect compromises one specific checkpoint.
But others are still working.
Exactly. Other checkpoints remain active, still trying to suppress that self-reactive clone. But, and this is crucial, over time, a particularly aggressive or high-affinity self-reactive clone might stochastically, just by chance, manage to evade the remaining functional checkpoints.
And maybe get help from other cells.
Yes, that increases the odds significantly. If you have, say, a T cell and a B cell that recognize different parts of the same self-protein, they can cooperate. The T cell helps the B cell make autoantibodies. The B cell presents antigen back to the T cell. This cooperation makes it much more likely they'll bypass the remaining tolerance mechanisms.
And that explains the variable onset, like why someone with a risk gene might not develop disease until middle age.
Precisely. It's the combination of the inherited susceptibility plus the random stochastic nature of bypassing those additional checkpoints. It makes the clinical onset unpredictable.
Okay, and then there's Model C, which involves somatic mutations.
Model C suggests that even without any inherited predisposition, a random somatic mutation could pop up within a single self-reactive T or B cell clone later in life.
A mutation affecting what?
Maybe something that disrupts its internal growth limits, or maybe it acquires a mutation that makes it resistant to apoptosis, perhaps affecting BCL-XL expression, or a checkpoint inhibitor pathway like CTLA-4. This random genetic hit within that one clone allows self-antigen to suddenly drive its expansion, again leading to that stochastic, unpredictable disease onset.
So in reality, it's probably often a mix. Inherited risk, plus maybe some somatic events, plus environmental triggers.
Almost certainly. It's rarely just one thing. It's a complex interplay.
Okay, let's put some specific gene names to these failure points. You mentioned PTPN22. Where does that fit in?
PTPN22 is a really important one, strongly associated with risk for type 1 diabetes and rheumatoid arthritis. It encodes a protein tyrosine phosphatase, basically an enzyme that dampens down signaling from the T-cell receptor.
So it acts like a brake?
Exactly. Certain variants of PTPN22 are thought to make this phosphatase less effective. The result is the brake isn't as strong, the T-cell receptor signaling isn't dampened as efficiently, and the T-cell becomes easier to activate. It essentially lowers the threshold for activation, making autoreactivity more likely. It's like adjusting the sensitivity knob.
Makes sense. Then there are the genes crucial for regulatory T-cells, TREGs, the immune system suppressors.
TREGs are absolutely central to peripheral tolerance. And here we look at genes like IL-2RA and CTLA-4.
IL-2RA encodes part of the IL-2 receptor, right? CD25.
That's the one. IL-2 is absolutely vital for TREG survival, function, and maintaining their suppressive identity. Variants in IL-2RA are strongly linked to both T1D and multiple sclerosis risk.
How do the risk variants affect TREGs?
Well, the T1D risk variant, for instance, seems to decrease the expression of the IL-2 receptor alpha chain, CD25, on the surface of TREGs. Less receptor means less efficient IL-2 signaling. This likely compromises TREG fitness, their numbers, maybe their suppressive capacity, weakening overall tolerance.
And CTLA-4, that's an inhibitory receptor TREGs use.
Correct. CTLA-4 is a key molecule TREGs use to put the brakes on other T cells, often by competing for those B7 costimulatory ligands we talked about earlier. Some autoimmunity-associated variants of CTLA-4 involve alternative splicing, meaning they affect how the gene's message is processed. Some research suggests these variants might reduce the production of a soluble form of CTLA-4. This soluble form might normally act as a decoy, soaking up B7 molecules in the environment. Less soluble CTLA-4 could mean less peripheral suppression, more effector T-cell activation.
And the ultimate proof of TREG importance comes from mutations in FOXP3 itself. This really highlights the foundational work of researchers like Sakaguchi, Brunkow, and Ramsdell in identifying this lineage, doesn't it?
Oh, absolutely. Their pioneering work establishing FOXP3 as the master regulator for TREG development was revolutionary. And the clinical consequence of losing FOXP3 is devastating. Mutations in this gene cause IPEX syndrome.
Immune dysregulation, polyendocrinopathy, enteropathy, X-linked syndrome.
Exactly. Without functional FOXP3, individuals essentially lack TREGs entirely. The work from Sakaguchi, Brunkow, Ramsdell, and others showed us just how critical this single factor is. The result in IPEX is catastrophic, uncontrolled T-cell activation against multiple self-antigens, leading to severe multi-organ autoimmunity, often fatal in early childhood if untreated.
So these rare Mendelian diseases like IPEX or ALPS from FAS mutations, they really serve as stark examples. They show us the extreme consequences when these key tolerance checkpoints completely fail.
Precisely. They provide clear, high-impact illustrations of the very same pathways that are likely failing, perhaps more subtly or incompletely, in the common, complex autoimmune diseases we manage daily. Understanding the role of FOXP3, thanks to that foundational research, is key to understanding tolerance.
Okay, let's pivot now to the cytokine milieu, interleukin-6, IL-6. Our sources paint this as a real central player involved in seemingly everything from chronic arthritis to acute infections.
IL-6 is incredibly important, yes, partly because it's produced by so many different cell types, not just immune cells like macrophages and T cells, but also fibroblasts, endothelial cells, you name it. And its signaling is unique.
Unique how? It uses the JAK-STAT pathway, like many cytokines.
It does, but the receptor system is key. IL-6 binds to the IL-6 receptor, IL-6R. Then this complex recruits two molecules of another protein called GP130, which actually does the signaling.
Okay.
But here's the twist. IL-6R exists in two forms. Membrane-bound on cells like hepatocytes and some immune cells, that's classic signaling. But there's also a soluble form, ACell-L6R, floating around in the blood. IL-6 can bind to the soluble receptor.
And then what?
And that complex, IL-6 bound to soluble IL-6R, can then bind to GP130 on cells that don't have their own membrane-bound IL-6R. This is called trans-signaling.
So it dramatically expands the range of cells IL-6 can act on.
Hugely, because GP130 is expressed almost ubiquitously. So trans-signaling allows IL-6 to have widespread systemic effects, driving inflammation far beyond the initial site. This is thought to be the main driver of its role in chronic inflammation.
Fascinating. Okay, let's focus on T cells. How does IL-6 shape T cell differentiation? It seems critical for pushing towards pathogenic responses.
It really is a key decision maker, especially for the Th17 lineage. To generate and maintain those pathogenic Th17 cells, the ones implicated in many autoimmune diseases like, say, experimental autoimmune myocarditis or models of MS, you generally need IL-6. Often it works together with other cytokines like TGF-beta and IL-23.
Th17 cells are defined by the transcription factor ROR-gamma-T.
Correct. IL-6 signaling promotes the expression of ROR-gamma-T, driving cells down that Th17 path. But it does something else simultaneously.
What's that?
It actively blocks the induction of regulatory T cells. Specifically, IL-6 inhibits the ability of TGF-beta to drive expression of FOXP3, the master regulator for TREGs.
So it pushes towards Th17 pathogenicity and slams the door on induced TREG generation.
Exactly. It's a double whammy. It actively steers the immune response away from regulation and tolerance towards sustained inflammation. That's why targeting IL-6 is so effective in many inflammatory conditions.
But you mentioned a necessary regulatory role too, something about the gut. That seems contradictory.
It does, but context is everything in immunology. While IL-6 generally promotes inflammation in the periphery, it appears to be required for inducing a specific type of regulatory T cell, these ROR gamma positive TREGs that are crucial for maintaining peace and quiet in the gut lamina propria.
Why there specifically?
Well, the gut is constantly exposed to trillions of commensal bacteria. You need specialized TREGs there to prevent unwanted inflammation against this microbiota. These ROR gamma plus TREGs seem to fill that niche, and their development depends on IL-6. So blocking IL-6 systemically could potentially disrupt gut homeostasis. It highlights the complexity. The same cytokine can be pro-inflammatory systemically, but locally protective.
Okay, very nuanced. Let's bring this to the clinic. Rheumatoid arthritis. IL-6 is a known bad actor there.
Oh, absolutely. In RA, IL-6 fuels the fire in multiple ways. It stimulates B cells, promoting autoantibody production, think rheumatoid factor, and anti-CCP. antibodies. It drives that Th17 Treg imbalance we just discussed, favoring inflammation.
And the joint damage itself.
IL-6 contributes directly. It disrupts the normal balance of bone remodeling. It cranks up the activity of osteoclasts, the cells that break down bone, and dials down the activity of osteoblasts, the cells that build bone. The net result is bone erosion and cartilage destruction, hallmarks of RA.
And the systemic symptoms. Fatigue, anemia.
IL-6 is a major driver of those two. It's the primary inducer of the acute phase response in the liver. That means increased production of proteins like C-reactive protein, CRP, serum amyloid A. These contribute to the systemic inflammation that causes things like fatigue, malaise, and the anemia of chronic disease. Clinically, serum IL-6 levels correlate very well with disease activity and severity, not just in RA, but also in conditions like giant cell arteritis and systemic sclerosis.
This understanding is now being linked to specific genetic risks, even in organ-specific diseases like vitiligo. Tell us about the IL-6 polymorphism study.
Right. This was a study looking at a specific single nucleotide polymorphism, an SNP, in the IL-6 gene promoter region, NASH 572GC. They compared vitiligo patients to controls. They found a pretty strong association. Individuals with the GG genotype had about a 2.5-fold increased risk of developing vitiligo compared to those with the CC genotype. The G allele itself was significantly more common in the patient group, carrying about a 2.4-fold increased risk.
Suggesting the G allele leads to higher IL-6 production or activity.
That's the implication, yes, that this common genetic variation influences IL-6 levels in a way that predisposes to this specific autoimmune attack on melanocytes. Conversely, the C allele seemed protective. It's a neat example of how a general inflammatory mediator can be linked genetically to a very specific organ-targeted autoimmunity.
And this deep understanding of IL-6 is absolutely critical now in acute care, especially managing cytokine release syndrome, CRS.
Yeah, IL-6 emerged as arguably the central cytokine in CRS across different contexts. During the COVID-19 pandemic, high IL-6 levels quickly became recognized as a major biomarker, predicting severity, ICU admission, and mortality. It was clearly implicated in driving that hyperinflammatory cytokine storm leading to ARDS and multi-organ failure.
And this knowledge directly informed treatment for CAR T-cell therapy toxicities.
Absolutely. CRS is a well-known, potentially lethal toxicity of CAR T-cell therapy. It typically hits 3 to 5 days after infusion, caused by this massive release of cytokines from the activated SAR and T cells and other immune cells they recruit.
Symptoms being fever, low blood pressure, low oxygen.
Severe symptoms, yes. And the standard of care, first-line treatment, is tocilizumab.
The anti-IL-6 receptor antibody.
Exactly. By blocking IL-6 from binding its receptor, you effectively shut down that systemic inflammatory cascade driven by IL-6 transsignaling. It can dramatically and rapidly reverse the life-threatening symptoms of CRS. It's a fantastic example of targeted immunotherapy based on mechanistic understanding.
What about the neurotoxicity, ICANS, that can also occur with CAR T? Does IL-6 play a role there too?
Immune effector cell-associated neurotoxicity syndrome, ICANS, usually develops a bit later, maybe 5 to 7 days out. The primary treatment for ICANS itself is usually high-dose corticosteroids like dexamethasone.
But tocilizumab might be used too.
Often, yes, especially if the patient has significant CRS concurrently, which is common. While steroids are key for the neuroinflammation, controlling the systemic IL-6-driven inflammation with tocilizumab is often necessary as well. The idea is that the systemic cytokine storm likely contributes to the breakdown of the blood-brain barrier and fuels the neurotoxicity. So hitting both IL-6 and using steroids is a common strategy.
Speaking of CAR T toxicities, our sources flag another emerging issue: thrombotic risk, blood clots.
Yes, this is something clinicians managing CAR T patients need to be very aware of. Thrombosis isn't just a background risk. It seems to be a direct consequence of the therapy in some patients.
How common is it?
One multicenter retrospective study cited in our sources found thrombotic events in about 7.1% of adult CAR T-cell recipients. That's not insignificant. These were mostly venous events, pulmonary emboli, PEs, deep vein thromboses, DVTs, though arterial clots occurred too.
And what predicts who's at higher risk?
The analysis found 2 key factors significantly associated with developing a clot after CAR T infusion: the peak level of D-dimer, which is a marker of clot formation and breakdown, and the severity grade of ICANS, the neurotoxicity.
So higher D-dimer and worse neurotoxicity correlate with clot risk.
Exactly. It suggests that the intensity of the systemic inflammation, and particularly the neuroinflammation, are driving activation of the coagulation system. This makes a strong case for considering thromboprophylaxis, especially in patients who develop severe CRS or any grade of ICANS, and for closely monitoring D-dimer levels.
Okay, let's shift gears again. Let's talk about how external factors, specifically viruses, can interact with our immune system and potentially trigger autoimmunity through molecular mimicry. SARS-CoV-2 obviously gave us a huge real-world laboratory for this.
Indeed. Molecular mimicry, the idea that a peptide sequence on a pathogen looks similar enough to a self-peptide to trigger cross-reactive T cells or antibodies, it's a long-standing hypothesis for how infections can initiate autoimmunity.
And with SARS-CoV-2, especially early on with vaccines using the whole spike protein, there were concerns.
There were, yes. The worry was that generating a very strong immune response against a large, complex viral protein like spike might inadvertently generate clones that also recognized human tissues just due to chance sequence similarities.
Do we have data on that sequence overlap? I saw something interesting about animal models.
Yes. Analyses were done comparing short peptide sequences, hexa and heptapeptides, from the SARS-CoV-2 spike protein to mammalian proteomes. They found significant overlap with human proteins as expected.
But less so in monkeys.
Interestingly, yes. Rhesus macaques showed the lowest level of sequence similarity to the spike protein compared to other common lab animals. This might actually explain something.
Which is,
Why macaques haven't been great models for severe multisystem COVID-19 in humans. The hypothesis is that they lack some of the key human self-antigens that the spike protein mimics. Without those shared sequences, they don't develop the same kind of cross-reactive immune responses and the associated autoimmune-like pathology that might contribute to severe disease in humans.
that's a really clever insight into model limitations. But what about direct evidence in humans? Did antibodies against SARS-CoV-2 actually cross-react with human tissues?
The evidence here is pretty striking and, frankly, a bit concerning. One study took human monoclonal antibodies, highly specific antibodies generated against either the spike or the nuclear protein of SARS-CoV-2, and tested them against a panel of 55 different human tissue antigens.
And the result?
Significant reactivity was found against 28 out of the 55 human antigens. That's more than half.
Wow, what kind of tissues?
A broad range, including important barrier proteins, components of the gastrointestinal tract, thyroid antigens, and even neural tissues. This provides a very plausible molecular basis for some of the persistent multisystem symptoms seen in long COVID, and also for how the infection could potentially trigger de novo autoimmune diseases in susceptible people.
Were there any specific hotspots identified, particular self-antigens that showed strong mimicry?
Yes. The spike protein showed notable similarity between 50% and nearly 80% identity at the epitope level to mitochondrial M2 protein.
Mitochondrial protein that could link to fatigue.
Exactly. It offers a potential mechanism for the profound fatigue and potential mitochondrial dysfunction reported by many long COVID patients. Another specific hit was the viral nucleoprotein showing similarity to TPO, thyroid peroxidase.
TPO is the main autoantigen in Hashimoto's thyroiditis.
Precisely. So this mimicry strongly suggests SARS-CoV-2 infection could be a trigger for autoimmune thyroid disease.
Okay, now let's bring in the genetics of the host, HLA type. How does that influence susceptibility versus the potential for autoimmunity after infection?
HLA molecules are critical because they're the proteins that present peptide fragments from viruses or from self to T cells. HLA class I presents shorter peptides, mainly to CD8 plus killer T cells. HLA class II presents longer peptides, mainly to CD4 plus helper T cells. Which HLA types you inherit determines which peptides you can present effectively.
And there was a study in Italy linking specific HLA types to COVID-19 risk.
Yes, an important study looked at HLA class I alleles and the incidence of SARS-CoV-2 infection in different Italian regions.
Which alleles stood out?
They found that HLA-CR1 and HLA-B44 were positively associated with a higher incidence of infection.
Meaning people with those alleles were more likely to get infected. What's the proposed reason?
The hypothesis is that these particular HLA molecules might be relatively non-proficient at presenting key SARS-CoV-2 peptides needed to generate a strong protective CD8 plus T cell response early on. If you can't efficiently display the viral danger signals to your killer T cells, the virus might replicate more easily or persist longer.
Okay, that makes sense. But what's the autoimmune connection? You mentioned these alleles might be risky.
Here's the really interesting part. Both HLA-B44 and HLA-CO1, the ones associated with higher infection rates in that study, also have known associations with various inflammatory autoimmune diseases.
Ah, so the same HLA types that might be poor at fighting the virus are also linked to autoimmunity.
Exactly. It suggests these HLA variants might be generally prone to mounting suboptimal or somehow inappropriate immune responses, whether the trigger is a virus or a self-antigen. They represent a kind of inherent genetic vulnerability on the HLA front.
Okay, let's look at the aftermath post-vaccination autoimmunity. There was specific data on vitiligo risk after COVID vaccination.
Yes, a large cohort study conducted in South Korea provided some compelling real-world evidence. They compared the incidence of new-onset vitiligo in people who received COVID-19 vaccines versus those who remained unvaccinated.
And the finding?
They observed a significantly higher cumulative incidence of vitiligo in the vaccinated group. By 3 months post-vaccination, the incidence was 2.2% in the vaccinated group compared to only 0.6% in the unvaccinated group.
That's a noticeable difference.
It is. The authors concluded that vitiligo should be considered a significant, though rare, potential autoimmune adverse event following COVID-19 vaccination.
Did the type of vaccine matter? Or the schedule?
Yes, intriguingly, the risk wasn't uniform. The highest risk for developing vitiligo was seen in individuals who received a heterologous vaccination schedule, meaning they got different types of vaccines for their doses, like maybe an adovector followed by an mRNA.
What was the hazard ratio for that group?
The HR for heterologous vaccination was 3.89. This was notably higher than for those who received only cDNA-based vaccines like AstraZeneca or J&J, where the HR was 2.86, or those who received only mRNA vaccines, Pfizer-Moderna, where the HR was 2.47.
So mixing vaccine types seemed to carry the highest risk for this specific outcome.
In this study, yes. It suggests that the specific way the immune system is stimulated, perhaps the type and duration of inflammation generated by different platforms or combinations, might play a role in triggering this particular autoimmune response in susceptible individuals.
Okay, our sources also delve into some really advanced tools and specialized areas of autoimmunity. Let's start with humanized mouse models. Why are these so essential now?
Well, standard lab mice are great for many things, but they often fall short when you're trying to study uniquely human immune responses, especially things involving HLA molecules, which are so different between species or human-specific pathogens.
So humanized mice are.
They're typically strains of mice, like NSG, NOG, NRG, or the human mouse model mentioned, that are severely immunocompromised. They're genetically engineered to lack critical components of their own immune system. Often they have mutations in genes like RAG2, so they can't make their own T and B cells, maybe before and knocked out, so they lack cytotoxic cells. And usually the IL-2 receptor gamma chain is gone, which knocks out NK cells and affects cytokine signaling.
Clearing the decks, basically.
Exactly. You create an immunological vacuum. Then you can engraft human immune cells, often peripheral blood mononuclear cells, PBMCs, or hematopoietic stem cells, into these mice. The human cells take root and establish a functional human immune system within the mouse.
And how well does that human immune system work in the mouse environment?
Surprisingly well, according to the studies. For instance, human PBMCs transferred into the Humapia mice model were shown to mount robust antibody responses. When these mice were vaccinated with hepatitis B surface antigen, HBsA, the engrafted human B cells produced significant levels of human IgM and IgG, specifically against the vaccine antigen.
So they're useful for testing vaccines.
Yes, and for studying human immune responses to infections, evaluating immunotherapies, understanding transplant rejection, autoimmunity, anything where having that human HLA context and human immune cell repertoire is critical.
And you can even make them specific for certain HLA types.
Absolutely. Researchers have created transgenic mice that express specific human HLA alleles, like the HLA A11DR1 transgenic mice mentioned. These mice have antigen-presenting cells that display peptides using those particular human HLA molecules.
Allowing you to study T-cell responses restricted by those HLAs.
Precisely. It allows for the precise mapping of T-cell epitopes, these specific peptide fragments recognized by CD8 plus CTLs or CD4 plus T helper cells for pathogens like Ebola or MERS-CoV, but restricted by the human HLA molecules you're interested in. It's a powerful tool for dissecting those HLA-restricted mechanisms.
Okay, circling back to CAR T-cell therapy and thrombosis. We know the incidence is around 7%, and it correlates with D-dimer and ICANS severity. What does this mean clinically?
It means thrombosis needs to be actively monitored for and potentially prevented. That 7.14% figure is a significant clinical signal. The fact that it's mainly venous thromboembolism, VTE, and linked to the severity of inflammation, high D-dimer, bad ICANS, points towards an inflammation-driven prothrombotic state endothelial activation, coagulation cascade activation.
So prophylaxis might be warranted.
It strongly suggests considering VTE prophylaxis, especially for patients experiencing significant CRS or any grade of ICANS, and closely tracking D-dimer levels could help identify those at highest risk. It also implies that effectively controlling the underlying inflammation with drugs like tocilizumab or steroids might have a dual benefit, reducing clarsicans and potentially lowering the thrombosis risk.
Okay, let's shift to a very different specialized area, folate receptor autoantibodies, FRAA, in autism spectrum disorder, ASD.
Yes, this is a really intriguing intersection of neurobiology and autoimmunity. Several studies have documented the presence of autoantibodies targeting the folate receptor alpha protein in a subset of children with ASD.
And this receptor is important for getting folate into the brain.
Critically important. Folate receptor alpha is the main transporter for getting folate across the blood-brain barrier. If you have autoantibodies blocking this receptor, you can develop a condition called cerebral folate deficiency, CFD. Even if your blood folate levels are normal, the transport into the brain is impaired.
An autoimmune mechanism causing a specific neurological nutrient deficiency.
Exactly. And what's compelling is the therapeutic data.
The leucovorin study.
Right. A double-blind, placebo-controlled trial looked at treating ASD children with DL-leucovorin calcium, which is a form of folate that can bypass the block receptor to some extent.
And what did they find?
They found significant improvements, particularly in verbal communication, in the children who received leucovorin compared to placebo. But the effect was dramatically stronger in the subgroup of children who actually tested positive for those folate receptor autoantibodies, the FRAA positive group.
How much stronger?
The number needed to treat, or NNT, for improvement in verbal communication in that FRAA positive group was just 1.8. That's a very low NNT, indicating a strong treatment effect in that specific subgroup.
A powerful signal for targeted therapy. Is there a deeper mechanism proposed beyond just repleting folate, something about mitochondria?
Yes, that's a leading hypothesis. Leucovorin, specifically the DL form used, is known to accumulate readily within mitochondria, and it's actually the preferred folate form for supporting mitochondrial function.
Yeah, mitochondrial dysfunction is common in ASD.
It's increasingly recognized as a common feature in a significant subset of individuals with ASD. So the thinking is that leucovorin might be improving symptoms, especially high-energy demand functions like verbal communication, partly by boosting mitochondrial health and energy production in neurons.
Fascinating. Okay, one last specialized area. This one is complex and historically charged.
Yeah.
Gulf War illness, GWI, the anthrax vaccine, and the role of HLA genetics in protection.
Right. The potential link between the anthrax vaccine, which contains the PA or protective antigen, and the development of GWI has been debated for decades. A key question has always been, why did only some vaccinated veterans develop GWI symptoms while others remained healthy?
The implication being it wasn't just the vaccine itself, but how an individual's immune system handled it.
Exactly. And a study cited in our sources provides really strong immunogenetic evidence for this. They compared the HLA class II profiles of Gulf War veterans who received the vaccine, comparing those who developed GWI versus those who did not.
And what did the HLA data show?
It showed a striking difference. They identified 18 specific HLA class II alleles that were present only in the vaccinated veterans who remained healthy, GWI negative. These 18 alleles were completely absent in the group that developed GWI.
18 protective alleles. What was the hypothesis about why they were protective?
The hypothesis was that these specific HLA class II molecules must be very good at binding and presenting fragments of the PA antigen from the vaccine. Efficient presentation to CD4 plus T helper cells is crucial for generating a strong, effective antibody response and properly clearing the antigen.
Did they check if these alleles actually bind the PA antigen well?
They did, using in silico prediction methods, and the results were clear. Every single one of those 18 protective HLA alleles was predicted to have a very high binding affinity for the PA antigen, typically a percentile rank below 1, meaning they're among the absolute strongest binders for that antigen.
So the genetics strongly suggest protection comes from efficient antigen presentation.
That's the compelling conclusion. If a veteran had one of these high-affinity HLA alleles, their immune system could effectively see the PA antigen, mount a robust CD4 plus T-cell response, get good B-cell help, make high-affinity antibodies, and essentially clear or neutralize the vaccine antigen efficiently without triggering a problematic chronic response.
And if they lack those protective alleles?
If they lacked these high-affinity binders, their immune system might have struggled to process the PA antigen effectively. This could lead to a weaker or dysregulated immune response, perhaps persistent antigen or inflammation, ultimately contributing to the complex constellation of symptoms we call GWI. It really shifts the focus to the individual's genetic capacity to handle the specific exposure.
So wrapping this up, we've journeyed through some incredibly complex, interconnected territory today. From the very fine-tuned molecular switches like NFB and BCL-XL that dictate T-cell fate,
Right, the fundamental checks and balances of tolerance.
to the broad systemic impact of a cytokine like IL-6, playing roles in everything from RA and vitiligo to acute CRS and COVID or after CAR T therapy.
And then seeing how external factors like SARS-CoV-2 can exploit molecular mimicry, potentially triggering autoimmunity by resembling self-antigens like mitochondrial proteins or TPO.
And tying it all together is the individual's genetic landscape, particularly HLA type, which we saw can determine susceptibility to infection, but also, remarkably, provide protection against vaccine-associated illness like GWI if you have the right high-affinity alleles.
It really underscores that these complex inflammatory diseases and autoimmune conditions aren't usually caused by one single thing. It's almost always a multifactorial failure, a breakdown in multiple checkpoints, influenced by genetics, maybe triggered by infections or other environmental factors.
And for physicians listening, understanding these intricate dynamics is becoming increasingly crucial, wouldn't you say?
Absolutely. Our therapies are becoming more targeted, using tocilizumab for CRS or maybe leucovorin for FRAA-positive ASD. These interventions work because they hit specific molecular pathways we've discussed. And recognizing that individual genetic makeup, like those protective HLA alleles in GWI, can fundamentally alter disease risk or outcome. That's the direction personalized medicine needs to head.
We've laid out how autoimmunity emerges from this confluence of inherited risk, random somatic events, and environmental triggers. It leads to conditions where, within one diagnostic label, you might have multiple different underlying pathways active in different patients.
Which brings us back to the challenge of defining and understanding complex chronic syndromes, something medicine has wrestled with for a long time.
So maybe a final thought to leave our listeners with.
Given everything we've discussed, the multiple checkpoints, the role of stochastic events, the profound impact of individual genetics like HLA in determining protection versus disease, as shown so clearly in the GWI data, it really makes you question the pursuit of a single unifying mechanism for some of these incredibly complex multi-system illnesses. What are the inherent limitations of trying to find one cause or even one disease definition when the biologic reality, the very mechanisms of protection, seems so fundamentally individualized?